Gene regulation and expression. Think of a library filled with how-to books. Would you ever need to use all of the books at the same time? Of course not. Now picture a tiny bacterium that contains more than 4,000 genes. Most of its gene codes are for proteins that do everything from building cell walls to breaking down food. Do you think E. coli uses all 4,000 plus volumes in its genetic library at the same time? How are prokaryote genes regulated? DNA binding proteins in prokaryotes regulate genes by controlling transcription. Bacteria and other prokaryotes do not need to transcribe all of their genes at the same time. To conserve energy and resources, prokaryotes regulate their activities, producing only those genes necessary for the cell to function. For instance, by using enzymes found in the local environment instead of manufacturing them, the bacterium does not waste energy. DNA binding proteins in prokaryotes regulate genes by controlling transcription. Some of these regulating proteins help switch genes on, while others turn genes off. The genes in bacteria are organized into operons. An operon is a group of genes that are regulated together. The genes in an operon usually have related function. For example, the 4,288 genes that code for the proteins in E. coli include a cluster of 3 genes that must be turned on together before the bacterium can use the sugar, lactose, as food. These 3 lactose genes in E. coli are called the lac operon. Lactose is a compound made up of 2 simple sugars, galactose and glucose. To use lactose for food, the bacterium must transport lactose against its cell membrane and then break the bond between glucose and galactose. These tasks are performed by proteins coded by the gene of the lac operon. If the bacterium grows in a medium where lactose is the only food source, it must transcribe these genes and produce these proteins. If grown on another food source, such as glucose, it would have no need for these proteins. The lac genes are turned off by proteins that bind to DNA and block transcription. On one side, the operon's 3 genes are 2 regulatory regions. The first is a promoter, P, which is a site where RNA polymerase can bind to begin transcription. The other region is called the operator, O, which is where DNA binding protein known as lac repressor can bind to DNA. Promoter, RNA polymerase binds here, and operator, repressor binds here. And here are the lac genes. Here's a lac repressor gene, codes for the repressor protein. When lactose is not present, the lac repressor binds the O region, blocking RNA polymerase from reaching the lac genes to begin transcription. The binding of the repressor protein switches the operon off by preventing the transcription of its genes. The lac repressor protein has a binding site for lactose. When lactose is present, it attaches to the lac repressor and changes the shape of the repressor protein in a way that causes it to fall off the operator. Lactose attaches to the operator and it disconnects so that it does not stop transcripting. When the repressor is no longer bound to the O site, the RNA polymerase can bind to the promoter and transcribe the genes of the operon. In the presence of lactose, lactose attaches and it releases so that the RNA polymerase can continue to transcribe. How are genes regulated in eukaryotic cells? By binding DNA sequences in regulatory regions of eukaryotic genes, transcription factors control the expression of those genes. One interesting feature of a typical eukaryotic gene is the TATA box, a short region of DNA containing the sequence TATATA or TATAAA that is usually found just before a gene. The TATA box binds a protein that helps position RNA polymerase by marking a joint just before the beginning of a gene. So the TA box is here. RNA polymerase. Transcription factors form a binding site for RNA polymerase. Gene expression in eukaryotic cells can be regulated at a number of levels. DNA binding proteins known as transcription factors regulate gene expression at the transcription level. By binding DNA sequences in the regulatory regions of eukaryotic genes, transcription factors control the expression of those genes. Some transcription factors enhance transcription by opening up tightly packed chromatin. Others help attract RNA polymerase. Still others block access to certain genes. In most cases, multiple transcription factors must bind before RNA polymerase is able to attach to the promoter region and start transcription. Gene promoters have multiple binding sites for transcription factors, each of which can influence transcription. Certain factors activate many genes at once, dramatically changing patterns of gene expression in the cell. Other factors form only a response to chemical signals. Eukaryotic gene expression can also be regulated by many other factors, including the exit of mRNA molecules from the nucleus, the stability of mRNA, and even breakdown of the gene's protein products. Why is gene regulation in eukaryotes more complex than in prokaryotes? Cell specialization requires genetic specialization. Yet all of the cells in a multicellular organism carry the same genetic code in their nucleus. Complex gene regulation in eukaryotes is what makes specialization possible. For years, biologists wondered why cells contain lots of small RNA molecules, only a few dozen bases long, and don't belong to any of the major groups of RNA, mRNA, tRNA, or rRNA. These small RNA molecules play a powerful role in regulating gene expression by interfering with mRNA. After they are produced by transcription, the small interfering RNA molecules fold into double standard hairpin loops. An enzyme called the dicer enzyme cuts or dices these double-stranded loops into microRNA, miRNA, each about 20 base pair in length. The two strands of the loops then separate. So larger RNA molecule, here's a dicer enzyme. Here's the miRNA, the microRNA. Here's the mRNA. And here's a silencing complex, matching the complementary base sequence to miRNA. One of the miRNA pieces attaches to a cluster of proteins to form what is known as a silencing complex. The silencing complex binds to and destroys any mRNA containing a sequence that is complementary to miRNA. So the silencing complex will not allow translation of this portion that matches the miRNA. The miRNA sticks to a certain mRNA molecule and stops them from passing on their protein making instructions. So this will stop and no protein is produced. Blocking gene expression by means of miRNA silencing complex is known as RNA interference, RNAi. So no translation is RNAi. At first, RNA interference, RNAi, seems to be a rare event found only in a few plants and other species. It is now clear that RNA interference is found throughout the living world and that it even plays a role in human growth and development. The discovery of RNAi has made it possible for researchers to switch genes on and off at will, simply by inserting double-stranded RNA into cells. The dicer enzyme then cuts this RNA into miRNA, which activates silencing complexes. These complexes block the expression of genes producing mRNA, complementary to the miRNA. RNAi technology is a powerful way to study gene expression in the laboratory. It also holds the promise of allowing medical science to turn off the expression of genes from viruses and cancer cells. It may provide new ways to treat and perhaps even cure diseases. So, what controls the development of cells and tissues in multicellular organisms? Master control genes are like switches that trigger particular patterns of development and differentiation in cells and tissue. Regulating gene expression is especially important in shaping the way multicellular organisms develop. Each of the specialized cells found in the adult originates from the same fertilized egg cell. As an embryo develops, different sets of genes are regulated by transcription factors and repressors. Gene regulation helps cells undergo differentiation, becoming specialized in structure and function. Edward B. Lewis was the first to show that a specific group of genes controls the identities of body parts in the embryo of a common fruit fly. Lewis found that a mutation in one of these genes actually resulted in a fly with a leg growing out of its head in a place of an antenna. From Lewis' work, it became clear that a set of master control genes, known as homeotic genes, regulates organs that develop in specific parts of the body. Molecular studies in homeotic genes show that they share a very similar 130 base DNA sequence, which was given the name homeobox. Homeobox genes come from transcription factors that activate their genes that are important in cell development and differentiation. Homeobox genes are expressed in certain regions of the body, and they determine factors like the presence of wings or legs. In flies, a group of homeobox genes known as Hox genes are located side by side in a single cluster. Hox genes determine the identities of each segment of a fly's body. They are arranged in the exact order in which they are expressed, from the anterior to the posterior. So, anterior, pink, green, yellow, and blue, posterior. Pink, green, yellow, blue, expressed in the embryo, expressed in the adult fly in the same order. In this figure, the colored areas of the fly show the approximate body areas affected by genes of the corresponding colors. A mutation in one of these genes can completely change the organs that develop in specific parts of the body. Clusters of Hox genes exist in the DNA of other animals, including this mouse shown, and in humans. These genes are arranged in the same way, from head to tail. The colored areas of the mouse show the approximate body areas affected by genes of the corresponding colors. The function of Hox genes in other animals seems to be almost the same as it is in fruit flies. They tell the cells of the body how to differentiate as the body grows. So mouse chromosomes, multiple chromosomes, they match. Head to tail, anterior to posterior, anterior to posterior, and the gene coding matches. Nearly all animals, from flies to mammals, share the same basic tools for building the different body parts. Master control genes, genes that control development, are like switches that trigger particular patterns of development and differentiation in cells and tissues. In prokaryotes and eukaryotes, environmental factors like temperature, salinity, and nutrient availability can influence gene expression. For example, the lac operon in E. coli is switched on only when lactose is the only food source in the bacteria's environment. Metamorphosis is another example of how organisms can modify gene expression in response to their environment. Metamorphosis involves a series of transformations from one life stage to another, such as the transformation of a tadpole to an adult bullfrog. It is typically regulated by a number of external, environmental, and internal, hormonal factors. As organisms move from the larval to adult stages, their body cells differentiate to form new organs. At the same time, old organs are lost through cell death, recycling nutrients. For example, under less than ideal conditions like drying pond or a high density of predators or a low amount of food, tadpoles may speed up their metamorphosis to frogs. The speed of metamorphosis is determined by various environmental changes that are translated into hormonal changes, with the hormones functioning at the molecular level.